All right, let's talk about one of the most fundamental processes in all of life, cell division. I mean, every single living thing, from a tiny little bacterium to you and me, we all exist because of this. It's the engine that drives life, growth, and reproduction. The goal? It's always the same. Take one parent cell and make two perfect, identical copies. But as we're about to see, nature came up with two very, very different ways to get the job done. I mean, it sounds simple enough, doesn't it? One cell splits, now there are two. But, oh, the methods behind this magic are where the real story is. Because while the outcome seems simple, the journey to get there is this incredible marvel of evolutionary engineering. And it was solved in two brilliantly different ways. So, let's meet our two main players in this story. First up, you've got the prokaryotes. Think bacteria. They're using the original, ancient blueprint for division. It's super streamlined, incredibly efficient, and crazy fast. Then you've got the eukaryotes. That's our team. Plants, animals, us. We developed a much more complex, sort of highly regulated upgrade to handle our much more complicated internal setup. Okay, so let's jump right in with that original method. We're going to take a look at how prokaryotes pull off cell division with a process that is just the definition of elegant efficiency. Seriously, this system has been working like a charm for billions of years. The process is called binary fission, and the absolute key to why it's so simple and so fast is a huge difference you can see right here. Prokaryotes don't have a nucleus. Their genetic material, just one single circular chromosome, is just kind of floating around in a region we call the nucleoid. No pesky nucleus, no internal membranes to deal with, and that one fact changes everything. And when you break it down, the process is it's so logical. First, the cell makes a copy of its DNA loop. While that's happening, the cell itself starts to get longer, stretching out and pulling those two copies of DNA to opposite ends. Then this special protein ring snaps into place right in the middle. This ring acts like a drawstring bag, pinching inward, building a new wall, and then pop, it splits the cell cleanly into two identical new bacteria. And that special protein I just mentioned, yeah, it's a total rock star. It really deserves its own moment in the spotlight. Meet FZZ. You can think of this single protein as the master architect of prokaryotic division. It's the director of the whole show. So what does this little protein actually do? Well, FTZ is the one that builds that critical ring right in the cell's midsection. It's what coordinates the split, and it even recruits all the building materials needed for the new membrane and cell wall. It's basically an all-in-one division machine, and here's a little teaser for you. This ancient protein has a modern-day descendant doing a very similar job in our own cells. Just keep that in the back of your mind. So that brings us to the other side of the family tree. Us, the eukaryotes. You know, as cells got more complex, as they started packing their DNA inside a nucleus and having a lot more of it, that simple pinch-in-the-middle trick from binary fission, it just wasn't going to cut it anymore. New challenges meant we needed a whole new solution. And this lays out the hurdles perfectly. A eukaryotic cell, first off, has to deal with a nucleus. It's like a vault for the DNA. You can't just pinch it in half. And instead of one neat little DNA loop, you've got multiple long, stringy chromosomes that have to be sorted perfectly. You can't have one new cell getting shortchanged. With all this extra complexity, you need a seriously sophisticated internal organizational system, a kind of microscopic machinery to pull the whole thing off without a hitch. So how in the world did eukaryotes solve this massive organizational problem? This right here is where the story gets really, really cool. It turns out nature didn't invent a totally new tool from scratch. Nope. Instead, it repurposed that ancient one we just met. It's a tale of two proteins, separated by billions of years of evolution. So here they are. On the left, you've got FTZ, the prokaryotic ancestor. And on the right, its direct descendant in eukaryotes, a protein called tubulin. Now, tubulin is the fundamental building block of the complex machinery that meticulously separates chromosomes in our cells. And look, this isn't just a fun theory, it's a cornerstone of cell biology. As the source material puts it so well, FTZ and tubulin are homologous structures derived from common evolutionary origins. What that means is they share a common ancestor. Evolution literally took the original FTZ blueprint and tinkered with it, adapted it for a brand new, much more complex job inside the eukaryotic cell. And I mean, the family resemblance is just undeniable. Both of these proteins can build themselves into long filaments and ring-like structures. They both use the exact same energy molecule, GTP, to power their work. but their jobs have specialized over time. FTZ, remember, pinches the cell from the outside. But tubulin? It builds this incredible internal scaffolding, the mitotic spindle, that grabs onto the duplicated chromosomes and pulls them apart inside the cell before it even thinks about dividing.
Okay, so let's put this all together. Let's do a direct side-by-side comparison, kind of like a scorecard for these two amazing approaches to making new cells. Just look at the contrast here. Prokaryotes, one simple loop of DNA, no nucleus. They can use that FTZ ring and divide, boom, super fast. But eukaryotes have all these chromosomes tucked away inside a nucleus that requires a whole separate process called karyokinesis, using that complex mitotic spindle just to sort out the DNA before the cell itself can split. The whole thing is slower, yeah, but it's unbelievably precise.
So here's the big takeaway, the really crucial point. It's so easy to look at this and think, oh, the eukaryotic way is more advanced, it's better. But that's really not the right way to think about it. Binary fission isn't inferior. It's the perfect, high-speed solution for a simpler kind of cell. Eukaryotic mitosis isn't necessarily better. It's the necessary adaptation required to manage a much more complex internal world. Each one is a winner in its own context.
And that kind of leaves us with this one last big, juicy question to chew on. If that simple, fast, and incredibly efficient method works so well, it allows bacteria to multiply at mind-boggling speeds, then why did evolution ever go down this long, complicated road to complexity? What was the big payoff? What did eukaryotes gain that was worth giving up all that incredible speed? Well, that's a story for another day. Thanks for joining me.